Okay, ready for part two now. And part two, the back page of my handout, it's just going to be continued down with your guided notes. Step 5, basically, is the analysis part of our experiment. Okay, in the scientific method, we have worked our way through. Move myself over here a little bit. We've worked our way through this experiment. This is where we have our independent variable, our dependent variable, all of our constants. Okay, we gather our data, and we figure out what it means. All right, all of this I'm grouping as analysis in our notes. Okay, so we're trying to establish what it is that what we're working through, and then we'll build that into a conclusion. So that's going to be step 6 of our notes. All right. So, oops, that's not what I wanted to do. Still trying to get into the swing of things here. It takes a little while for a teacher to get some rhythm. All right, so we're in the analysis section, step 5. And when we gather the data, one of the things that we're going to notice about our plants, besides what we're measuring, okay, we're measuring height, and that, since we're using centimeters or inches or feet or whatever it is that we're working with, that's a type of quantitative data. Okay, quantitative data is comprised of numbers, of measurements, of actual data. Okay, so numbers would work here too. But we'll also be paying attention to how green the leaves are, how big the marigold flower is, if it's a flower, if we're flowering, how lush it looks, how strong it looks, if it's all withered. Okay, so that's qualitative data. That's just a description of what it is that we're working with. So these are important, just as important as the data. Okay, they don't necessarily go in a data table, but they still need to be written down. These are what we call observations. Okay, so this qualitative data are observations, and we want to make sure that with our observations that we're also providing information about what's going to happen. In a chemistry lab, if you mix two things together and you get some kind of blue powder, okay, start floating around in the liquid, that's not something that you're measuring, but it's important to know what happens when you mix those two things together, right? Okay, so that would be our qualitative data. So we're paying attention to our plants. Excuse me. We're paying attention to our plants with how they look as well, what the soil looks like. Okay, and we're comparing all four, even with the control. We're looking at it as well. The results of the experiment, and this is typically the quantitative data, will end up in a data table. Or some people call it a data chart. Okay, I'll call it a data table. And it organizes things for you. If you're a reader and you're reading over somebody else's experiments, you don't necessarily want to read everything word for word. You're looking for an introduction about what it is they're trying to do, the purpose. Okay, you're looking for the results, and that's what you're going to look for in the results. Then is this data table. Only if you're going to try to repeat the experiment will you read the procedure necessarily, or if you're curious about how they got the results. Okay, what did they do? Okay, but the first thing you look at is the organized data. All right, so once we have this data table, the next thing that we might do with it, and this is appropriate sometimes, but not appropriate at other times. You won't do this for every lab that we do if we get to have in-person labs. All right, but we want to graph the data. Oftentimes we'll graph it. We'll want to make a graph. And this is just, of course, you all know what a graph is, but it's a picture of the data. It's a picture view of data. And what it allows us to do is to begin to see trends, consistencies, things that the pattern that it's making. All right. Now, in some classes, when you graph, there'll be a bar graph. That happens with biology a lot. Okay, especially if you're doing something like over time or something like that. okay, or year to year. Psychology, social studies, they might use a bar graph. And another versions of bar graphs, there's lots of different types of these bar graphs. You can put the same data into a circle graph. Okay, you can put the same type of data in what's called a line graph. Okay, now with a line graph, so a line graph, basically, if you've got these bars that you're not drawing in a line graph, and they're not necessarily going to be a consistent height, in a line graph, it's basically you're taking these points at the top and you're just connecting them. Okay, so it's going to go up and down. Now you see this a lot when you're doing analysis, like the data that they're collecting for COVID-19 is the number of cases in Kentucky, for example, every day of confirmed cases. And that's definitely a line graph. It's going all over the place, okay, like that. That's a line graph. They could have done the same thing with a bunch of bars next to each other. So since it's every day, you're getting this different type of data. Now, what we want to do in science, in chemistry at least, when we're doing a graph, is often we will do what's called a scatter plot. Okay, since we're doing a graphing independent versus dependent, and I'll show you how to graph later on when we do density. But if we'll do a scatter plot, and a scatter plot basically is an X versus Y type situation. Okay, so we'll have an X-axis, that's our independent variable. We'll have the Y-axis, which is our dependent variable. And on this axis, we'll plot the data points, how the X versus the Y occurred. Okay, and what we're looking for is we don't want to connect the dots, is we're looking for a trend. Okay, so what we would draw then is an average of the trend line, okay, that through as many of those points as possible. See if it's linear. It may not be linear, though. It may end up looking like this, okay, which we would draw a line then in this case that would be some kind of exponential curve, maybe, or it could be logarithmic. Okay, we don't know the relationship. That's what we're trying to establish when we do one of these graphs. So an XY scatter plot is the type of graph that we'll be working with mostly, okay, if we're gathering data. Now, for the take-home lab that you're going to be doing, you're going to be graphing your independent variable and dependent variable, and it would be okay to do it as a line graph, okay, or a bar graph. But if you're doing something that actually has a relationship, that's a mathematical relationship, then you probably would want to use a scatter plot of some type. Okay, so one of the tricks is to be able to pick the best type of graph. I'm just telling you that that's a main one. This is called a line graph, and this is called a scatter plot. All right, so we'll analyze it somehow. And maybe we just look at the data, or we do calculations. Calculations would be a part of this. Okay, so all of that happens in this results or data or analysis section. Once we've established our trend, or we've done our calculations, or we've done our measurements, and we've compared them, and we've looked at them, and we've processed them, the next thing we want to do is figure out what we learned. Okay, and that's what we call a conclusion. We write a conclusion. Now, a good conclusion needs to be in paragraph form. Okay, so that's important. So we want to make sure that we're following that. Proper rules of grammar, of course, full sentences. Any good conclusion, regardless of the format chosen, will at minimum include these main sections. Okay, so if you were taking notes, and you could be taking notes along with me side by side in a notebook or something, but if you're taking notes, I would have you star this section. There's two parts of the experiment that you're going to be doing at home, the investigation that you're going to be doing at home, that I'm going to be putting my most attention to when I grade it. One of those sections is step 6, this conclusion. The other is from part 1, the procedure. Okay, so make sure that you pay attention to these details, that I tried to emphasize how much detail you needed to put into it, etc. Okay, so those are the two main sections that I'm going to be focused on as I grade that assignment. That's one of my focuses this year, is to help you improve those two different sections. Now, if you don't have to write a procedure, that's good. That's okay. Somebody's already written it for you. You just make sure that you copy down or write down what you did. Okay, and that's the important part of in our labs, if we were actually performing the lab, is that we have our step-by-step instructions, but those step-by-step instructions may not be, you might want to write down what you're going to do, but then as you start doing it, you learn, hey, that's not working. Okay, I'm not having any luck with this step. Well, then adjust it, change it. Okay, so you come up with your plan, your procedure, and then you start working through your plan and you realize that's not actually what I'm doing this. So adjust your procedure to fit it before you send in the report. All right, so you have some play there. Now, the conclusion, of course, is after we've done the procedure, after we've gathered the data, after we've analyzed the data, after we've learned something, we start to write this conclusion. Was our hypothesis correct? Okay, so that's the first part, is review the problem. Okay, did ALA grow plants better or did it kill the plant? I don't know. Okay, did Mountain Dew, Diet Mountain Dew is what I drink. Did Mountain Dew work better? Did Coke work better? Did water work better? Okay, that's why we're doing the control. So maybe all of our plants all of our drinks hurt the plants except for the water. So there's a reason maybe that plants prefer water. So we want to restate that problem. Explain the reasoning behind the hypothesis and the procedure. Okay, now you're not doing all of these. Okay, when you write a conclusion, you're not going, okay, step 1, restate the problem. Step 2, explain the reasoning behind it. Okay, this is just to help you focus your paragraph 1. Okay, so if you restate the problem, you're also restating the hypothesis, probably. All right, so you just want to make sure that you include this information in some format. It's not a checklist. All right, the checklist is paragraph 1 needs to deal with the review of the problem. All right, that's step 1, is you need to tell the reader so that they don't have to go back to the beginning and read everything that you're doing to figure out why it is that you're doing it. This is telling you why, what the purpose was. Okay, so why we did the experiment. Paragraph 2, once you've talked about why we're doing this, what was the purpose of it. Step 2 is you want to review the data and match the data with the hypothesis, the purpose. So this is where you talk about if it worked. Okay, you want to talk about if your hypothesis was proven or your hypothesis was disproved. Okay, does the data support the hypothesis or the purpose? Describe what happened in the experiment. Support the description with actual data. Okay, discuss the data. Don't just point them to the data chart. Talk about the average height. Okay, L8 had this average. Coke had this average. Mountain Dew had this average. Water had this average. So as you can see from the average height after the 2-week period, or 1 week, or how much time we would have decided that we're going to measure this, do that dependent variable, is we want to support that, okay, with actual data. So numbers have to be included, both quantitative, talk about your results with your descriptive data too, your observations. You made your observations while you were gathering data, okay, but you shouldn't have to go read the whole report to find those. You want to summarize those in this section. Okay, so the conclusion is going to be a synopsis of what the lab was. You're not going to say how you did it. You're going to say why you did it, what you discovered, what your errors are, and what you learned. Okay, so that's the 4 steps. So review the data, then you're going to talk about your error. So if there were any of our constants that we couldn't maintain, okay, like flavor, color, okay, those could have potentially influenced the error. Maybe we forgot a day and we didn't feed it one day. Maybe we weren't adding it to the same part of the plant, okay, that we accidentally added one of them over to the side so it didn't get directly onto the roots. Okay, any of those kind of things. What flaws that you discovered in your experiment? What errors that you might have made? Were the results what you expected? Okay, if L8 killed the plant, maybe it was because it got drier faster. Okay, we don't know. What we're trying to do is we're trying to figure out what happened that went wrong. What could we have done better? All right. So we're looking for mistakes, and they might be egregious, like that blue powder that I said when you mix the two chemicals together, and you got this blue powder, and say we want to collect it, and we start pouring it through a filter, and we overfill the filter paper, okay, so that it goes down the sides and onto the counter. And that's hard to collect. It's hard to recollect. So we wouldn't have that blue powder then that we could measure later. That's an egregious type of mistake. All right, that's going to affect our results, our yield. All right, so that would be an error that you need to include. So those are the kinds of things that you want to discuss here. And then last but not least, we want to answer the question of what did we learn? Okay, what was it that we got out of this experiment? And it may be that it just points us toward further investigation, okay, that we didn't learn very much. All we learned was that L8 kills plants, and all sodas kill plants, maybe, is what we discovered, and that maybe we need to try fruit juice, okay, if we're trying to find something that's better, something that helps plants grow better. Or maybe we should have tried an error. Maybe we should have had water with the same amount of sugar in it that the other plant, so that we took that out of the equation. Okay, so we're trying to analyze and understand what it is that we did and what it is that we can do better and what the next step might be. Okay, what's the next step in this investigation? Maybe it would take more. We were only adding a cup. Maybe we should have been adding 2 cups every day. Okay, so those are the kinds of things that we need to think through as the next steps. 1 cup helped the plants grow. If we tried 2 cups the next time, that will have changed one of the variables. Okay, so at that point, the independent variable is still the different types of drinks, but we've increased one of the constants. And maybe we want to add different amounts do a different experiment that we add different amounts of each of them, or we use L8 for all of the plants in a different experiment, and we adjust how much we add for each one. So we take the best and we adjust how much. Okay, so if we're trying to figure out how to make plants better, there are lots of different directions we can go with this experiment. Okay, go for further investigation. So we'd want to explore those if we were scientists, if we were trying to understand a problem, what helps plants grow better. All right, so that's the experimental data, experimental design notes. So what you're going to do now is you're going to take the information that it's discussed here in the guided notes, and you're going to then answer some questions, okay, and a little short progress check, and start thinking about an investigation. All right, and I've got an investigation plan for you, so you can go ahead and look at that assignment. Thanks a lot.